So I think you will have lots of questions. We will also take questions from the floor. So especially if the last talk inspired you to discuss certain things, then you're welcome to ask questions. So more are coming in. I will try my best to read it. So I'm starting with a more general question for Claire. It was asked by someone who just found a natural product isolated from streptomyces. And the question was a little bit general. What would be the first steps to characterize its toxicology? How would you start when you identify a natural product?
So I would start in the literature, to be honest, and see if there's anything out there with a similar chemical structure or a similar use that may have information out there that gives you some clues. And then depending on how much compound you actually have, I would start, if we know nothing about this chemical class, because even though it's from a natural product, it is a chemical class, I would start looking at some early in vitro screening, things like just general cytotoxicity, what happens when it goes into a mammalian cell. Is there anything particularly surprising?
Thank you. Then one for Terry. It's about juvenile tox studies in rodents. How good of a surrogate is it for exposure liabilities in neonates? Is there any regulatory experience available?
That's a good question. In terms of surrogates, we don't have a lot of experience in our division with surrogates, because I said the bulk of our biologic products are actually the monoclonals, in which there are no good relevant animal models. But from other divisions where they do ask for surrogates, the surrogates requires a fairly extensive amount of research and development. And when they're used, the neonates is something that they really do want sponsors to look into, particularly when it comes to reproductive toxicology studies. And you would assume that perhaps if the surrogate is going to work in the adult animal, that there is some relevance in the neonates, but that should come out in the reproductive toxicology studies.
So maybe I continue with another question for Terry. Please elaborate on what you mean with identification, the wrong target organ. Sure. So maybe the person who asked this question just needs a little bit more clarification. Absolutely.
And it is true. When I mention target organs, I mean it's either a target or it's not a target. However, when we talk about dose-limiting toxicities or dose-setting toxicities, we tend to try to focus on those organs that are the most sensitive, and the ones that are, in particular, if there's a question about reversibility, or the ones that will cause the most problems to patients. So we tend to focus on things like CNS and liver toxicity, for instance, and we prefer to use those for the dose setting for the first-in-human trials.
I mean, I guess you get it wrong if you don't know what you're looking for and you don't look. But like the systolic blood pressure question, I asked our tox group, how did we not see this? And it's not a normal thing you look for. We do all the ECG work in dogs. We do your general observations, heart rate, all that, and stuff. So we did the standard package. That's not part of the standard package. So we missed that effect, right? And so it's one of these challenges. If you look at everything, you'll definitely see it. But what is everything? And I think that's an example where we missed something because it's not normal to look for.
And just to follow up too, it's, you know, we also tend to look at things in terms of the most sensitive species. So if you see a finding in one species and not another, and how do you sort of relate that to a sort of a clinical risk?
A question for Ryan. How does trimethoprim sulfonamide combination and tetracyclines fit into the inner membrane mammalian toxicity idea?
Yeah, I mean, the reason I kind of cheated and went to anti-pseudomonal is it narrows things so quickly, right? Basically, we have quinolones and aminoglycosides. Those are the only two things that work inside the cell there. That's why we're so committed to LPXC. I mean, I think if you look at the multi-gram dosing agents, which we were trying to be if you want to be an anti-pseudomonal with our LPXC program, it's generally not the case, right? That you can get enough drug on board. You're pretty limited on dose. But obviously, somewhere, and the quinolones, actually, someone should ask about that. I had a whole nother thing, but it takes too long. I have some thoughts on why they're unique, but I think they're kind of miracle that they can pull off what they can pull off. I think part of it is that so little drug needs to get inside to actually cause a catastrophe, whereas if it was a target like Heinz talked about earlier, where you need 99% inhibition, I bet the quinolones never would have seen the light of day. So a little bit of this mechanism, but I think in E. coli and Enterobacteriaceae, there's a couple examples where you can eke out enough selectivity, but you see those are lower dosed agents is the main thing, but fair point.
So staying on the outside of the bacterial cell is still a good target. But there's, I mean, not that much is happening in this space. It's still mainly penicillin binding proteins and not that much outside of this target.
Yeah, it's true. Well, and on the outside would be great, right? We had a biologics program for a long time trying to do things on the outside, and all we have is really the polymyxins, and that outer coating is so dynamic, and maybe it's this big separation with the gram-negatives and how we're failing so much compared to the gram-positives is just they've been a lot better about keeping things out. I mean, right, no ATP in the periplasm. It's in basically equilibrium. It's almost like a little like sieve. It's not even a real membrane. And there's not a lot of essential processes, and the ones that are kind of like these large protein-protein interactions. So programs we've thought about or looked at are disrupting large surface interactions, and the mutation rates are just through the roof. So you can theoretically make it work, but then you get mutations because it's big surface area interactions you're trying to disrupt. So that's where we're kind of stuck. I think you're right. There's some fatal flaws in all this that we're trying to think deeper, another layer down than the just single gene thing. That, you know, other first principles we've missed that are really limiting what we can do. But I haven't seen, Genentech had a neat program they had a talk on that I'm hopeful is targeting something in the periplasm that's essential, so we'll have to see. But for now, the only thing we know for sure we can count on is the PBPs, yeah.
Yeah, it seems to be so intriguing to identify a genetic target and then find an inhibitor. Seems logical, but it has lots of risks. Are there questions from the floor? Okay, then I'll try the next question. This time for Claire. With respect to early in vitro toxicity screening of new molecules, is there a consensus for a panel of tests that are broadly recognized as being the best indicators?
Good question, but in short answer, no. So there's lots of different assays out there, and it really comes down to maybe the question you're trying to ask. There are, in tox now, there's an awful lot more emphasis on trying to get target organ-specific assays, talking about organs on a chip and 3D models. The difficulty with those assays is they tend to be much more expensive. They use more compounds, and in my experience, they don't necessarily give you the whole picture. An example would be for nephrotoxins. So some people, obviously, certain classes of antibiotics are known to be nephrotoxic, so people consider using a kidney-specific panel to have a look at that. But there's been quite a lot of data out there in the literature showing that actually looking at cytotoxicity in a mammalian cell is all that's really going to give you, because for a nephrotoxin, the really important thing is the distribution of the drug. It's not just about activity against the cell, it's how long the drug is there on the cell and whether it accumulates in the cell. So really for the in vitro assays, I'd say just a general mammalian cell is your best bet. And then for me, the earliest indication you can get is when you get in vivo, whether that be for your efficacy studies, PK or tox studies. And the sooner you look in vivo, the sooner you'll have an understanding of any real effects.
Another question. Assume a drug candidate which suffers, for instance, from a species-specific metabolism limiting exposure in rodents more than tenfold. How do you obtain tox data in 2 species to support the IND and phase 1 studies?
I can.
Yes.
So one thing is you have to explore every rodent species. So I have been involved in programs where we looked in hamster. We have looked in ferret. You have to explore all possible rodent species before you can state that you can get no exposure. There are also ways of trying to, depending on your route of administration, is there any way you can overcome the clearance by increasing the drug load, even if that's not your intended route as you go forward? Is it a way to just increase the exposure in the animals for the toxicity testing?
And also, we know in many cases the rats tend to chew up the drugs quite rapidly. Their half-lives tend to be much faster than humans. So, I mean, if you have sort of like proof of concept preliminary studies in another, like a non-rodent species, I mean, those are going to be the ones that we're going to rely on the most anyways. An animal that doesn't, you can't reach similar exposures in wouldn't have a lot of value for us, FDA.
I have to admit I have problems with reading the handwriting. There seems to be a difference between how Europeans and Americans learn in school the handwriting. So for me, it's like like reading Egyptian hieroglyphs. Maybe I'll ask my colleagues here if they're better.
So there's one question here which we think is saying, is there rules of thumb about toxicity limits of small molecules? And we're not sure if you're thinking. So in terms of a preclinical package, the guidance is that you have to achieve one of 3 things. You either have to be dosing at your maximum tolerated dose, or you have to be dosing at your maximal feasible dose. And if you're at a maximum feasible dose in rodents and non-rodents, that's at 1,000 mg per kg per day. Except if you think in the clinic that you may be at over 1 gram a day, and then you actually have to be at 2,000 mg per kg in the rodent. And if you haven't achieved a maximal tox dose or a maximal feasible dose, the other option, which in my experience doesn't really work for anti-infectives, is you have to be at 50-fold your predicted efficacious dose. But for most anti-infectives, that's not going to be applicable.
I've another one for Claire. For cytotoxicity, which cell lines are recommended? Can you comment on use of cancer cell lines versus progenitor blood cells?
Yeah, so it's always a question really, because obviously a cancer cell line, this is a non-normal cell, and however they're useful because it makes it easier to run screens. It's very difficult to run screens on a primary cell line. In my experience, we used to use things like A549s or the TK6 cells. These are both cancer cell lines, but it allows you to use it in a screening mode. Again, as your program progresses, and if you are concerned about cytotoxicity, then that's something where you maybe would progress into a more organ-specific model and then start looking at things like a primary cell line. But again, if it's just early on, you're just trying to understand your risks. How toxic is your compound? Are you killing these cells at nanomolar potencies? And if so, that's going to be a cause of concern.
So this is for you, Ursula, but I can read it to you. So what insights about entry are available from the IMI translocation program?
Oh.
I figured.
Yeah, I mean, I'm not member of this project, so I cannot really answer that. What I've heard is it's now ending already, and they have lots of information, detailed information. I'm not sure yet if anyone has somehow summarized it to a kind of guidance what to improve in drug discovery. So I'm still waiting for that. But please go to the website of Translocation, and I'm sure that at least they have hundreds of publications listed there. But I still would like to see a summary of all these studies.
So the question is about the how long it takes to do some of these testing, non-clinical testing. The question is, I think a 1-month GOP toxicology studies has taken 5 to 6 months to complete, start to finish. Then you have 1-month, 28-day studies with 28-day recoveries, 2 to 3 month histopath and reporting. Is there any way to go faster? And then what is the role of in silico toxicology? So from our perspective, we generally, when someone comes in with an IND, we typically want a report that's been QA'd. It doesn't have to be necessarily in the final form, but it would have all the data, particularly the histopathology report that we can review. We understand that these studies do take time, but keep in mind also that you also don't have to necessarily do a 1-month study in an animal to support a 14-day indication in humans. You can go to a 14-day study in animals as well. In terms of in silico toxicology, we're not at a stage right now to really support that sort of in lieu of, or to accept that in lieu of the animal testing and the animal data. It is supportive, but we really do need to see those animal studies at the point.
I'll just add, having been through a bunch of these campaigns, like it seems like it takes a long time, but it's usually CMC holding you up anyway, so it might not be a big deal. So see John's CMC bootcamp from last year's meeting, because I think some of those understandings might make you think differently about whether or not you need to hurry.
I think also, taking on that CMC point as well, I think it's also about the planning. So when you do your preclinical package, there's a number of steps before you get into those pivotal studies, even if they're 14 days. You have to do some dose range setting, some finding work to make sure you're at the right doses for those GLP studies. And so when you put that all on a Gantt chart and you put on your CMC, actually you can run a lot of these things in parallel. There are obviously some risks because you may have a toxicity that means you can't go forward, but it's the way to shorten that program length.
We have one question about immune responses, but we are not quite sure if maybe.
I'll give it a shot. So immune responses to large molecules were a significant feature of several cases presented at the recent FDA non-traditional workshop and also the one today. Are there general rules for predicting, and I believe it's dealing with these? I think anytime you take a particularly humanized animal or a humanized biologic and you put it into animal species, you're going to potentially see some kind of a immune response. The question that we're primarily interested in is whether this affects PK, is it neutralizing, non-neutralizing, and is there any real toxicities associated with that immune response? I mean, clearly you have to choose your models. Rodents may be more sensitive to that than, say, a cynomolgus macaque. So you have to sort of find the most relevant species and then sort of rationalize and justify sort of the findings that you see.
In my experience, well, for biologicals, there are in vitro models to try and understand whether you're overstimulated, because for adversity, the main risk is overstimulating the immune system. And so there are, after obviously we've had some issues in the clinic with these molecules, there are assays available to understand your risk of human hypersensitivity of immune response, looking at things like PBMCs, etc.
What about biomarkers for toxicity in early phases? So could you comment on that?
So I can give my... Okay. So in my experience, unfortunately in toxicity, a lot of our biomarkers are really a response of effect. So you can see changes in things like liver enzymes or your kidney markers, but actually that it's not really, for me, it's not a true biomarker. It's not showing you the effect before the change. It's actually showing you a level of damage that has occurred, and you then are measuring it. There aren't, in my experience, a lot of biomarkers that show you the effect before damage has really occurred, which is really what you're looking would be ideal, really, because then that means you could stop your dosing before you start seeing that toxicity. In terms of using those in earlier programs, again, if you know you have or you think you may potentially have a risk of particular target organs, there are particular circulating biomarkers you can take to address that target organ. Troponins for the heart system, there are kidney markers, there are liver markers. So again, it's trying to identify early on what you think the potential hazards might be, and then you can use any in vivo assays you have, any PK studies, any efficacy studies, any tox studies, to try and understand that risk a bit better.
What just came to my mind, are there any non-traditional models for assessing some specific toxicology processes that you would recommend for very specific questions? So not the routine models.
So it's difficult. So I don't think there's anything routine you would use in that sense. I think there are obviously earlier markers, things like cardiovascular effects. So traditionally, obviously, we need to use a non-rodent for first time in man, but you would use, say, a guinea pig for QT effects early on. You'd maybe use a rat telemetry to understand blood pressure effects and heart rate changes early on. But a lot of you do have to think about what that question is and designing the studies to suit it.
And from a regulatory perspective, I think that if you were to present or come in with a non-traditional animal model, it may be difficult for us to really understand that model if we have no experience with it. I think there's value, and again, it's supportive and mechanistic, and we would definitely like to see it and review it, but I think in general, the more traditional animal models, ones that we have more experience with, would be easier for us to understand.
Now a very difficult question. What is number 1 and number 2 mysteries seen in preclinical tox programs? Is this? Mistakes. Oh, mistakes. Not mysteries. Thank you. My reading. I thought a very interesting question, but mistakes makes better sense.
When they get to you.
Sure, I can start.
Maybe you find also some mysteries in this.
I think the primary ones we see is the exposures, that the animals aren't adequately exposed to cover the clinical exposures. As I mentioned, a lot of times, particularly rats, they burn through the drug very fast, they clear it very fast, and so a 30-day study or 28-day study in a rat doesn't necessarily equate to a 28-day study in the clinic. A lot of times, or vice versa, sometimes you need longer studies in animals. So it's just sort of, that's probably one of the mistakes we see, is that we really, you have to take into account the half-life, the PK, the similarities and differences between the species to really understand how to dose those animals.
Trying to separate it into like the mixture of known classes we've worked on and then novel stuff. So the known classes, you kind of go in knowing what you're looking for, hope you don't see anything new. But for a novel, it's always about, and this is maybe not your candidate, this is like as you're deciding, do you even have a lead? But it's like, what free peak can you achieve without causing harm? And the harm would almost always be cardiovascular. There's other things, but that'd be the thing you'd look for. The hard part is the right way to look is like in a dog or something where it's a really heavy duty study to pay for and to scale for. But what's your free peak that's tolerated and what's your free exposure that's tolerated? And if it's really big, you feel really confident. The one other issue with the phlebitis, which is worth mentioning, is I sort of struggled with, how do we miss, the potential for that in humans? And the data I had showed previous at a different setting, it was pretty severe. I mean, we got about 10 doses in before we saw significant, but you could see migration of like swelling and edema. And I think one of the challenges there was when you do your repeat dose toxicology, what are you doing? You're doing a central implanted catheter because you can't do a peripheral vein dose on a rat 3 times a day for 14 days. And probably somebody tell me the equation for allometrically scaling a rat central vein to a human peripheral vein. I don't think that's a known phenomenon. Like we were talking about like diameter, flow rate, all these things, and there probably isn't a great way. And I think going to a peripheral model with a gram-negative drug where you're dumping in, grams per day would be way wiser, and I'll definitely make sure we do that with the next program, because it wasn't going to bear out in the standard GLP repeat dose studies in the same mathematical way it would bear out in a peripheral vein.
Are there any questions from the floor? Here's one, please.
Just following up on this, for the exposure. So simply because you just said exposure might be one of the main mistakes, what would be the closest estimate from a regulatory perspective? You would not look at doses, you would not look at perhaps dosing intervals, you have a higher clearance. Would cumulative AUC come close, or how would you judge on the exposure being addressed appropriately?
Yeah, that's a good question. You know, cumulative AUC is something we have looked at in the past. I think it is a very difficult, I guess, thing to sort of understand. You know, when we first start out with the first in human dose, and clearly you don't have the human AUC, so you're going to do multiples. But at the end of the day, we clearly would need some kind of matching AUC to really be able to sort of understand the potential toxicities and risks associated. So I know some people have done longer infusions where you may not get Cmax, but you may get AUC for longer infusions. So it's really come down to, and we do see that occasionally. But if you believe that all the toxicity is related to Cmax, it's something that you might be giving up with the long AUCs. But eventually, perhaps if you went to another species, you went to a non-rodent species particularly, you may be able to start to see exposures that better match. We're looking for something that's at least clinically relevant. Best case scenario, it's at least 10 times for safety.
I guess one thing too, just thinking preclinically, since you sort of, you don't know your answer in people yet, and you're trying to estimate efficacy, for LPXC, our target was actually the greater of either, our PKPD target, the exposure, the greater of either what was required for a 2-log kill reduction in a model or 24 times the static exposure, which the answer was it was the second one. And part of that reason was we knew with Pseudomonas we're going to have MEX-CD mutants. We know we have to dialyze into the lung through the interstitium like any other drug, a gram-negative drug. And we just felt like if we really were going to be successful, that's what it would require. And I think I saw Paul Ambrose here earlier. I think he's the first person to open my eyes many, many years ago to the idea that it's just a given fact that your drug will have incredibly variable pharmacokinetics when you get to sick patients, especially in the pseudomonal world. And we just inflate all of that out of the gate. We're doing target attainment models with preclinical leads because we have data from plasmamycin, but we're able to plug it in and actually make estimates on how much do we have to overexpose just to account for the fact that when we give patients even weight-based doses in severely ill settings, the PK is going to be all over the place. So definitely have some headroom, otherwise you'll get to phase 2A or phase 3 and you'll be toast.
Or you test only in mild infections.
Yeah, or just treat the, I don't know, cystitis.
Exactly. If there are no more questions, then we'll come to our last round. So would you have one recommendation to the audience, one that you think is very important, specifically important to go home with?
I think probably one of the most important things is to come in and talk to us. I think that in regards to that other question about the number one mistakes, I think a lot of times sponsors come in with too much. If sponsors come in with a lot of studies or they're proposing to do a lot of studies, we're not going to say necessarily don't do them because we don't fully grasp the entire development program. But I think a lot of the programs can be streamlined to a minimum number of studies and types of studies. And I think that if you come in with the pre-IND consultation, we can sort of tell you which, if you ask those kinds of questions, which pivotal ones we're really interested in seeing. And I think that will sort of help expedite sort of your program.
So for me, it kind of links onto that actually. It's think about safety as early in your program as you can, both in the discovery bit and in your early development. The earlier you think about it, the more planning you can put in place.
That bought me some time. I mean, I can't really distill like something specific to tox to one thing from a program because it depends on what you're working with. But I guess the one thing that Terry made me think of is like, don't hesitate to talk to, if you're kind of newer in the industry or at a smaller company that hasn't been in this space, like don't hesitate to talk to the people from companies. They're really passionate about this stuff and they'll tell you stories and they'll share data and they'll be transparent. Not everybody, but like don't assume that because we're at companies we're like going to be super secret about everything we're doing. It's quite the opposite, and it's a super small community for those of us that all know each other over so many years. So if there's something you have an idea about, like, oh, I think I'm in good shape, but am I? There's plenty of ways in small molecule world to talk about your science without revealing your structure, which is really all that matters in our world. So like, just don't hesitate. People will help, has been my experience.
So I have seen a lot of big grant proposals and projects, and so my impression, my advice would be, don't just look at MIC values and consider everything that's above 8 as great, because if I read such results, I always think you really will need great PK and you really will need great safety data, otherwise it will not work. And this is very hard to achieve. So always integrate very early, integrate efficacy, safety, and PK, of course. Yeah, we are coming now to the end of this webinar. I would please ask you to give us your wish list for the next topics, topics for next year's bootcamp, but also topics for the webinars on the GARDP website. And if you have more questions related to these topics today, then you also can go on the revive.org website and just post your question there in the forum, and we'll find experts who will try to answer your questions. So there are really a lot of options for you to ask questions to experts on the revive.org website. So with this, also in the name of CARB-X and the other organizations who have supported this session, I would like to thank our speakers, and we'll end this session. Thank you.